I thought I'd follow up on this question. You had I'm not going to do this as a clicker question. I'm following up on a clicker question from last time. A student asked me, well, if you consider just the stop sign without the post, would this be an example of radial symmetry? And then if, in fact, you include the sign with the post, would this be an example of bilateral? Yes, that was an excellent observation. Absolutely, that's true. Hopefully that makes sense to everybody, right? Another thing I wanted to show you, I'm not sure if this is going to work or not. It's being a little funky with the changing the source. Yes, it worked. So, this morning's paper. I'm reading the local section, and there on the front page of the local section is an article about some recent fossils that were uncovered during what? Construction of a nearby highway. And so this is actually in the San Juan Capistrano area. And so what they have found, let's see, they have just found two old leatherback turtle fossils that are millions of years old. The part of the sediments where they're digging through the heavy machinery right now, they're estimating that those sediments were once about 3,000 feet below below sea level. So there's an interesting article, and I'd be happy to show that to any of you afterwards if you're interested. I had discussed before when we were talking about fossils that many of them are uncovered during construction projects because that's when rock layers are disturbed in a good way. And this is just another example of that. So let's go ahead and dive into the phyla that we'll be talking about today, starting with phylum Platyhelminthes, the flatworms. Some students have told me that as we go through the different taxa, they're having a hard time keeping track of different attributes. I think the easiest way is to recognize that these attributes form suites of interrelated characteristics, and so there are certain consequences of a particular kind of lifestyle, whether it's parasitic versus free living or aquatic versus terrestrial, that have a number of accompanying features. And so hopefully as we go through some of the taxa today, and also as you review some of the taxa you've already learned about, you'll see some of these common patterns and it will make more conceptual sense to you. But anyway, the flatworms may or may not be a familiar group to you. Perhaps as we go through some of the examples, they will be more familiar. If any of you are fans of the artist Escher, then you have probably noticed creatures like this crawling around in some of his magnificent geometric landscapes. He was apparently fond of the one particular kind of flatworm, which has this characteristic appearance, as you can see right here. And we'll look in just a little bit at some flatworms that look remarkably like this. But to go back to figure from your textbook then, this is figure 33-10 from your textbook, and the flatworms are featured right here in this middle panel. So flatworms are quite flat, and also significantly, they do not have a coelom, so no coelom. There is an interesting discussion in your textbook about the role of body cavities in locomotion. I think this can be a little hard to wrap your mind around if you're not used to thinking about locomotion and force generation in an animal that doesn't have hard parts. I think it's makes a lot more conceptual sense to consider movement when you're contracting muscles that attach to hard parts like bones. We move different parts of our bony skeleton by our muscle contraction. But something like a worm, then worms have muscles. It makes intuitive sense that as muscle cells contract and generate forces that could change shape. But how does this actually generate forces during locomotion? Well, one of the issues is that about a coelom specifically is that a coelom can be a cavity, can be a fluid compartment, rather, to push against, meaning that as muscles contract and generate a force, if there is a coelom, water is not very compressible, So water is not very compressible, which means what? That it transmits force very effectively instead of just simply going into a smaller volume, which is what it would do if it was compressible, but it's not, so it just sends the push on, so transmits force effectively. And so this is the basis for hydraulic systems, for example. So what that means then, in worms that do have a coelom or a pseudo coelom, as the worms both above and below the flatworms in this particular diagram, then they do have a coelom, which means that worms of that type, when they do contract their muscles, then this increases the pressure in the coelom, which can transmit forces elsewhere in the body where that coelom extends. Flatworms don't do this. This doesn't mean that they can't transmit forces. It just means that as the muscles contract, they're squeezing muscle cell, the rest of the body, rather than a coelom itself. So if you consider the body, actually I think green will show up fine. So a muscle that is oriented in this direction, for example, and shortens would cause a change in shape that would cause a shortening in that direction. A muscle that was in this orientation would cause further flattening and so on. So it probably makes intuitive sense to you that as the flatworm contracts its muscles, this changes the body shape. So how do flatworms locomote? Well, they use a combination of, let's see, flatworm locomotion. They use a combination of both beating their cilia, which they're covered with cilia, but also muscular contraction. And you'll see this when we look at some movies in just a minute. The other way in which a coelom is significant in terms of the body is as a sort of a rudimentary circulatory system, meaning that it is a fluid-filled compartment in which nutrients, for example, can be distributed in the body. So flatworms do not have that. So then an issue, of course, for flatworms in the absence of a coelom is, well, how are nutrients, for example, moved around inside the body? And there's a couple of different ways, of course, that this can be affected or caused. And one of the issues with being very flat in your body form is that all of your cells are pretty close to the outside. So this means exchange with the outside is facilitated, but also the flatworms. There's now a picture on the left-hand side of the screen that's being projected in front of you. This is not from your textbook, but I thought it was a helpful diagram of the basic anatomy of a flatworm. At least this is of a kind that's a so-called free-living, which means not parasitic. So this looks a lot like the ones that are featured in Escher artwork, for example. So the digestive system is shown in here. One of the things I'd like to bring to your attention first is this structure that's sticking out of the middle of the body is not a reproductive organ. It's actually it is where the mouth is on the base of a muscular tube called the pharynx. So this is the opening to the digestive system, and the digestive system, in fact, goes in an anterior and posterior direction as shown here. So food is then transported within the digestive system. So that's how nutrients are transported around in the body. This is an example of an organism with a so-called incomplete digestive system, meaning there is only one opening. So rather than being one long tube in which there's two openings and the digestive system runs from mouth to anus, there's just one opening. So food goes in this one opening, is distributed around, and then undigested material comes out the same opening. So this functions as both the mouth and the anus. So this is really different from many other animals. Let's see, what else can I point out here? You can also see the relatively simple nervous system shown here. These lines here are part of the nervous system, and then up at the front here, what gives this flatworm sort of a cross-eyed look are a couple of light-sensitive organs at the anterior edge of the head. These are not image-forming, but they're light-sensitive. So flatworms do not have a coelom, but if we look at the phylogenetic tree in front of you right now, figure 33-2 from your textbook, then you can see that this loss of a coelom is something that occurred in the evolutionary history of this particular lineage, because in fact the flatworms are derived from other organisms that do in fact have a proper cilium or body cavity. So this is an evolutionary loss over time. So let's go to some movies here. This is it's a little darker. There we go. So you can see this. So I think it's easier to understand the nature of some of these animals seeing them in actual motion. So one thing we consider is, well, where would you find some flatworms if you went out into the world and wanted to catch some? Well, one really easy way of doing this is just to get a jar of pond water with some vegetation in it. That's what's shown in front of you right now. And then after some time, if you check out your water, you may notice that there are a bunch of small worms up at the top there. And chances are they're flatworms. So these are all flatworms you're looking at here. They're transparent, many of them, so it's convenient. You can see their internal anatomy. This is the nervous system you're looking at here. Those two little black dots there are light-sensitive organs in this particular individual. So this is the anterior end. But remember, the mouth is not actually located on the anterior end. It's halfway down the body. So if we now watch a flatworm going after a meal, so here's a little tiny crustacean that will be lunch for this particular flatworm here. This is the anterior end. Let's see what happens when the flatworm comes near this little creature. First of all, it smears it with some mucus, and so that sort of has two effects on this little struggling prey item here. One, it makes it harder for it to move, but it also seems to be having a paralytic effect. So there are probably some chemical compounds in the mucus as well. So here's the little flatworm, and then its mouth and pharynx are right here, so it's sort of sucking the nutrients out of this individual, and then this is the remnants after the meal has been finished. So if an item is large like this, it's not actually taken into the digestive system. So let's look at this flatworm. This is a slightly different kind, as you can see from its appearance, and because of the close-up photography in this part, we'll be able to, it'll be very clear to you that what we're looking at is muscular contraction as it's moving. You can see that it becomes skinnier and fatter, and obviously then this is because the muscles are contracting. So there's a combination of muscular action and ciliary movements. So again, these are the light-sensitive organs. Now, many of these flatworms are carnivorous, as the ones that we're looking at, and so their eyes are not image-forming, but they often find their prey items via chemical compounds. So if you do something such as the individual who was making this movie did, put a flatworm in a dish of water with a little chunk of meat like this, then the flatworm can find it as following a chemical trail of chemical compounds that are just coming out of the food item and entering the water. This is the mouth at the end of the pharynx. It's going to take some of the nutrients out of that little chunk of food. So those were examples then of free-living flatworms. Not all flatworms are free-living. There are many that are parasitic, and one of the large groups of parasitic flatworms are a special group called the tapeworms, and there are thousands of different kinds of tapeworms. Some specialize in their hosts more than others, but this is an example of a kind that is found in humans. So let's go ahead and walk through this life cycle to understand a little bit more about this kind of flatworm. So let's start here with the adult. This is the adult tapeworm right here. Adult tapeworms, if they're found in a human host, are in the intestine. If they're in another kind of vertebrate or mammalian host, they're in that vertebrate's intestine. That's where adult tapeworms hang out. And they can be very, very long. I don't know if you can read the print on the screen in front of you, but it says that this is a particular kind is about 8 to 11 feet long typically. So that's very, very long. Of course, the intestines are also extremely long as well. One of the curious things about tapeworms is that they do not have mouths. They don't have a mouth. They don't even have a digestive system at all. And this may sound odd to you. No digestive tract, unlike the other flatworms we were just looking at, until you realize, well, after all, where are they located? They're inside a digestive tract, so they're completely surrounded and bathed in nutrients that are being broken down by digestive enzymes, and so the nutrients just move right across and absorbed right into the body walls. They can get by without a mouth at all. Now on the anterior or head end, they usually have specialized structures. This is not a mouth. These are little suckers. Usually at the head end of a tapeworm you have a combination of suckers or hooks, and the purpose of those is to remain attached to the intestinal wall of the host. So this is how this individual then stays in place and isn't dislodged as all the food and drink goes by in this person's as this person is eating. So what happens next? So this adult is hanging out in the intestine, and one of the remarkable things about tapeworms, you may be able to see that there are a number of different segments here. All of these segments have both male and female reproductive parts in all of these segments. So this leads to a tremendous reproductive capability of tapeworms. And so you have all these different segments capable of reproduction. And so if you think, well, how is this tapeworm supposed to sexually reproduce if it's the only tapeworm in the intestine? You know, no problem, because all of these segments have both male and female parts. Then one segment can just simply sexually reproduce with another one. Sperm can be exchanged between different segments, and the eggs fertilized, and there's no problem. If there are more than one tapeworm in the same intestine, then those worms can go ahead and reproduce with each other. Again, in fact, they can reproduce at multiple locations because they have multiple locations with both male and female reproductive parts. So after then the tapeworm has had an opportunity for sexual reproduction, then the eggs, or even some of the segments as a whole, are then shed. The rest of the adult worm stays in place and just sheds the fertilized eggs or these segments into the digestive tract, where they're eliminated along with feces, and that's how they then spread to other animals. And so, for example, this shows that if something like a pig or other livestock then ingests material that has been contaminated with feces from an individual that had a tapeworm infestation, then these eggs hatch out in the pig's intestinal system. But that doesn't mean that the pig then ends up with an adult tapeworm. It's a more complicated life cycle than that. What happens is that the larvae, after they hatch, then they burrow into the muscle tissue, and there they insist. I'm not saying the word insist. They're not demanding anything. They're insisting, meaning they are forming cysts. So the larvae then form these little cysts in the muscle tissue, and then they just wait. Nothing will happen unless that muscle tissue is then consumed by a carnivore. So, for example, this person is eating some pork. If this person cooks the pork thoroughly, the encysted larvae are killed off, the person's fine. If it's improperly cooked, then what happens? Well, the cysts then come out in the intestine, and then form adult tapeworms then in the adult. So if one has to have a tapeworm, then you really want to have the adult stage, not the encysting stage, because it's much less, it leads to far fewer medical complications or problems, because the adult tapeworms just simply stay in place in the intestine as opposed to ending up in muscle tissue. So in fact, people sometimes even infect themselves with tapeworms on purpose, and I've got an example of that, which is the next slide. I'll make it a little darker so you can see the movie here. So this is from this BBC News website, and this is Dr. Michael Mosley, who is a TV presenter, and so he's interested in how tapeworms affect the human body. So he deliberately infected himself. Let's see, where's that information? Right here. Yes, he swallowed some tapeworm cysts. Let's see, he had got those from, I believe, yes. He swallows 3 tapeworm cysts, which he obtained from infected cattle in Kenya. All right, and then after swallowing these, several weeks later, after swallowing the tapeworm cysts, then what did he swallow? He swallowed a little tiny spy camera. Okay, I'm scrolling down here in this movie so that I can show you what this looks like. So after swallowing the tapeworm cysts, he then swallows this little tiny camera. This little tiny camera will then go in his mouth, travel all the way down his digestive tract, come out the other end, but meanwhile, it's transmitting pictures to his iPad. Okay, so he's able to watch this on his iPad. So anyway, if we scroll back up here, then we can see the movie that is on this site as well. So this is then a picture of him watching the results on the iPad. You know, wow, look at that. Okay, I'm going to pause it for a second so I can point out things. So this is then images being transmitted from this camera that he swallowed, so it's in his intestine, right? So what we're looking at here, this is the wrinkly surface of the inside of his intestine. This is the tapeworm, right? See these different yellow segments. These are the different yellow segments that are capable of inter-reproducing. So anyway, oops, if we, let's watch the rest of this. Let's see, zoom, speed things up a little bit here. There we are. Okay, if we go back to where the movie starts, there we are. This is just take a few seconds, but then you can see, so this is what he can see on his iPad, right, little view into his intestine. And so there they are. He's got tapeworms right there. If you want to read about that, you can go to the PBS site. But anyway, fascinating though these flatworms are, we really do need to get through some of the other phyla. So let's go on to the next phylum, phylum Mollusca, the mollusks. The easiest way of understanding the mollusks is to recognize that the different types of mollusks are all modifications of a basic mollusk body plan, which is really quite simple. And here it is in front of you right now. This is figure 34-7 from your textbook, and shows the major parts of a mollusk. So there are basically just 3 parts here. There is an area called the muscular foot, and this is shown right here at the bottom of this typical molluscan body diagram. So this is a single structure that is muscular and is used in locomotion and is modified in the different groups, as we'll see in just a minute. Typically, there's just one, which is why it says foot and not feet. Then the second body part has the charming name of visceral mass, and so this just refers to a large chunk of the body that is just the internal viscera. So it's the digestive system, the reproductive system, just as one big chunk of the body. And then finally, we have a part of the body that is capable of secreting a shell, if in fact there is one, or otherwise could just form a part of the body that kind of wraps around another part of the body, and that's the structure way up here. So the purple thing right here is supposed to represent the shell for those mollusks that actually have one. Not all mollusks do. So let's go ahead and walk through some of the different representative groups. We'll start with the bivalves. This is a really easy name to wrap your mind around because bi means 2, valve just means 2 valves. These are the very familiar scalps and mussels and clams, so those mollusks that just have 2 parts. And so here's a picture of a bivalve, a real bivalve on the lower left, and then over here on the right is what you see if you pry one of the valves off. So these 2 valves are both made by the mantle then, so instead of just having a single shell, there are 2 different parts, and they're attached together by a hinge, which is right back here. So if we look at the generalized molluscan body plan here and match it up with the bivalve body, then you can see the different parts fairly readily. So here's the muscular foot here. Here's the foot over here. It's this part right here. So there still is a foot. It's still involved in locomotion. This will be a little easier for you to see in the movie than I'm just about to show you. Then we have the visceral mass, which is just the internal organs, and then a gill, if there is one, and that's just sort of the whole rest of the body here. The mantle that secretes the shell is just the part immediately next to the shell here. So very, very simple. The only other thing I want to point out here is a structure that is indicated at the anterior end of a mollusk, the so-called radula. This is a hard structure associated with the mouth and usually involved with feeding, as you'll see in some of the other mollusks, but there is no radula in the bivalves. And this relates directly to how they make their living. They're actually filter feeders. So these guys are all filter feeders. They make their living just as sponges or the lophophore-bearing lophotrochozoans, making their living from just collecting small organic particles out of out of the water. So how do they do this? Well, they bring in water. So this is shown in the lower right-hand part of this diagram. So water is brought in via the beating of a bunch of cilia that drive the water flow, and then water also comes out, and there are structures called siphons, which are just little rolled up tubes made out of the mantle tissue that underlies the valves. So let's now check out how they use their foot. So here's an example of a bivalve then, and so the two valves are separating from each other. This is time lapse. This is the foot then, right? So there's the foot, right? It's this muscular structure right here. And so bivalves use this for burying in the sand. It's a sort of remarkable thing to watch. There we go, it just dug itself underneath. And we'll see some more in just a second here. There are a couple of other bivalves. You can see them sort of moving around. They do use their foot. There's another one sort of burying itself. And so after they bury themselves, you often see this little tiny siphon sticking out. Why is that? It's because they still have to eat, right? They're buried in the sand, but this individual is then bringing in water and any organic particles so it can filter those out. That's how it's actually eating, and then it can expel the water. Let's see. Okay. So you may have noticed that these are items, clams, mussels, scallops. Many of these are items that you often see on restaurant menus, and so these are common food items ingested by humans. But when this is not a good idea is when those bivalves have been exposed to a so-called red tide. And I've got a picture of what a red tide looks like on the left-hand side here. Red tides are a name given to periods of time when part of the ocean experiences a population explosion of certain kinds of protists. And if those protists make defensive chemical compounds which are toxic to humans, then there are problems with eating bivalves that have been filtering out these protists, because of course these are plankton, and so these are exactly the kinds of things that are in fact eaten by these filter-feeding bivalves. So clams and mussels and scallops that are in areas undergoing a red tide are usually not fit for human consumption, and so fishermen have to look at shellfish safety information alerts such as this one. This is one for the state of Washington, for example, and so this shows different areas hatched with different colors showing where it's okay, where it's closed, where it's open, where is it okay to collect these bivalves and sell them for human consumption, when is it not. Okay, let's leave bivalves and go on to another big important group of mollusks, and that is the so-called gastropods, snails or slugs. And this is the group that's actually the easiest, I think, to match up with the typical molluscan body plan, which is still shown here on the left. And so there's a pretty close correspondence. The only difference, or the major difference, between the picture on the right of a snail showing some of its internal anatomy and the molluscan body plan shown over there on the left is the twist. And so what happens in most gastropods that have this kind of twisty shell is that the body itself is also has undergone torsion and has a twist as well. This is why, for example, the digestive system, which is shown here in hot pink, is shown as undergoing a loop because the body is twisted. But otherwise, it's really quite similar to this appearance of the mollusk on the molluscan body plan on the left here. So unlike the bivalves, which do not have a radula, the gastropods do, so the radula is right here at the front end, and so a snail, which is an herbivore then, will use this radula like a little scraping structure to feed. So we can see this in motion. Perhaps it would help to, sorry, this is going to do weird things, but this is acting a little buggy today. It's not cooperating. Okay, I'm sorry, it amplified, but it's not allowing me to enter the screen to turn this down. I'll have to just move this away so I'm not blasting your ears out. Anyway, So here's what you're looking at here is a snail crawling on the glass of a side of an aquarium. So this is where the radula is right here. Here's its mouth. This whole structure here is the foot. Let me rewind that. I'm sorry, I was distracted by the misbehavior of the electronic devices here. Let's see if I can just start it up again. So there's a single foot in this particular type of snail, the one that's being pictured here. You'll notice that the two halves are operating somewhat out of synchrony. You do see this in other snails. You just see the entire foot as a whole coordinated from left to right. So it's almost acting like a bipedal organism here, but it's just a single foot. But the radula, you can sort of see its action a little bit as it's scraping the glass, and this is how many snails and slugs make their living. They crawl around and they just scrape areas in their environment with their radula. So, but if you consider carnivores, they have modifications of the radula, and so a particularly interesting modification is seen in the so-called cone shells, which are some of the most dangerous, venomous gastropods in the world. They're also have beautiful, striking shells, and so humans that are in some of the Pacific Islands where these are more common see such gastropods, and of course they pick them up because they're very beautiful. This can be a problem because their radula has been transformed into a small harpoon-like structure which is like a poison dart. And they use this to shoot poison at their prey items. And so here's the movie. So what you're looking at here is a close-up of just part of a cone snail. So this is like the siphon area. You can see a little tube of the siphon coming out here. And it's primarily buried in the substrate here. They're not terribly visual. They're relying on chemical cues to find a particular prey item. Whoop, there's a fish. Not this fish's lucky day here. Okay, so here is then the siphon, and it smells the fish. Bam! Okay, just shot it with this little poison harpoon. These are neurotoxins that act. Whoops, there goes the fish. You see that? Kind of got sucked into the cone shell's body. So anyway, and now it's just sort of going under the surface here and will digest its fish meal. So many gastropods are sort of slow moving. Some of them are very dangerous predators. Okay, that's not quite big. All right. The last group of mollusks we're going to consider, so I'm not going to take time to go through the Polyplocophorans or any of the other ones. You are not responsible for knowing about the Polyplocophora. The cephalopods, these are perhaps the most familiar. These are the smart, large-brained, fast-moving organisms in the mollusks. And so they show different kinds of modification of their overall body plan than the other major groups of mollusks, but you can still see the same three parts. This is still figure 34-7 up at the top here, showing the overall molluscan body plan, and then down to the right is an example of a squid that is moving via jet propulsion. So this kind of means of generating locomotory forces is very unusual in the animals. And so to understand how the squid body relates to the overall molluscan plan, then the muscular foot is actually the part that is modified into the tentacles or arms of the cephalopods. The mantle is then this part right here that it sort of encloses the rest of the body, and you can't actually see the visceral mass because it's sort of wrapped around by the mantle. And so as this muscular part then contracts, it shoots water out of the cavity, which then sends the animal in the opposite direction. So this is how squids then move. It just started cooperating again. This is fantastic. Okay, I can make it dark again. Yay. So it'll be easier for you to see this movie, because now we're going to look at a really cool octopus movie. One of the other things that cephalopods are very famous for is their ability to change their characteristics, their color or texture of their of their body. This is a rather extraordinary film clip taken by Roger Hanlon. So he was filming underwater. This looks like a little chunk of seaweed or something until the octopus actually then changes color. They can do this extremely rapidly, and there it is, it's swimming off. Notice that cloud, that was not dirt being kicked up, that was ink, cephalopod ink being released by the octopus, which is now over here. Extraordinary, very, very unusual capability to change their shape. Now, here's another little clip to show you another aspect of octopus life. So this octopus has been put underwater in an enclosure with just a relatively small hole in it. Now, unlike the gastropods, many of which have these huge, large, clunky shells that they have to carry around, most cephalopods have very little in the way of any hard parts, and so this means that they have a very soft, squishy body which allows them to move through little tiny openings. So you can see that this octopus is pulling itself through a very small hole that is much smaller than its usual body size because it's just very soft and squishy. It doesn't have hard parts, so this allows an octopus to go into a crevice to get away from predators and also to go into a crevice after its own prey as well. And here's the last fun octopus video. This is showing a predator going after an octopus and the type of response that one sees. So this is not the octopus, this is a prey item. Whoop, there's the octopus releasing more ink. And so, of course, generating these huge clouds of ink is thought to be helpful in terms of hiding from the predator, which can no longer see it. There are also descriptions of cephalopods releasing ink in clouds that are sort of the same general size and shape as the cephalopod itself, and then changing color as if this is how they can hide. I don't know if that's really true or not, but anyway. Okay, we've gone through 2 phyla. Before we start the next one, let's go ahead and pause for a clicker question. So this is an example of a question that you should be able to answer based on what you're now learning about protostomes. So consider this. The common ancestor of the protostomes had a coelom, right? What does this suggest? A, that all Lophotrochozoans have a coelom. B, there are no pseudocoelomates within the protostomes. C, there are no acoelomates within the protostomes. Or D, the body cavity evolved before the Lophophore. So why don't you think about this for a while and consider what you think is the best answer. Okay, I'm doing this as an any answer counts clicker question. Just pick what you think is the most best answer for the question. Okay, has everybody clicked in? All right, let's see what you guys think. Interesting. I would say this is the best answer. This is actually sort of almost true, right? Although you have certainly seen a exception to this, haven't you? Namely, in the flatworms. So you know that the first one is not true, but certainly that would follow logically from this statement in the absence of having additional information about the flatworms. So I would consider either one of those just perfectly fine answers to this question. So nice job, everybody. So that's a bit, move this out of the way. Okay, so let's go now talk about the other major protostome group, the ecdysozoa. So when we consider the ecdysozoans, then we're considering the other group of protostomes, right? So again, just to remind you, here's a somewhat abbreviated version of the phylogenetic tree of the animals, and we're talking about the protostomes, right? We have considered a couple of different types of the Lophotrochozoans, and now we're going to turn our attention to the other group, the ecdysozoans. And So one of the major characteristics that is common with all of the different phyla within the Ecdysozoa is that these individuals are completely covered with a very tough and often very hard material called cuticle. And because this is tough and hard, this is related to the next point here, which is that during development it is typically molted. And this is because it is very hard to grow bigger if you're encased in something that is not terribly stretchy or flexible. And so usually these organisms then grow in a sort of stepwise fashion that is associated with their molting. The two largest phyla are the nematodes and the arthropods, and we're going to finish up on the nematodes today then. So let's dive into the nematodes. Here we are, right for you right now. There is a picture of a typical nematode, at least a free-living example, then on the left, excuse me, on the right, and then an example of what it looks like as they crawl around on the left, the movie. The common term for nematodes is roundworms. So this is in contrast to the flatworms, the Platyhelminthes that we've already discussed. There are so many different kinds of worms. So these are different from earthworms. They're different from flatworms. They're different from tapeworms, which of course you now know are a kind of flatworm. They're different from flukes and arrowworms and peanut worms and horsehair worms and ribbon worms and all the other worms that didn't even make it onto the list here. There are a lot of different kinds of worms. And initially taxonomists put all the worms together because after all being worms, they all share a number of traits. But it is now recognized that many of those different traits are in fact a result of convergent evolution, and many of them are not particularly closely related to each other. So, but what do we know about roundworms? There are many tens of thousands of species that have been named, and probably even more that have not yet been named. One of the difficulties with identifying and naming species is so many of them look exactly like this, okay? They don't have segmentation, they're very plain in appearance, at least the free-living ones. Habitats, often aquatic habitats, many live in soil. Some live in animal or plant tissues. If they live in animal or plant tissues, then they're parasites, not free-living. And in contrast to the flatworms that we have already covered, these ones do have, in fact, a complete digestive system. You can kind of see this as this worm moves by. You can sort of see some of its internal anatomy, meaning that there is a mouth on one end, and then you have a long digestive tract, and then at the other end you have the anus. So it is in fact a complete digestive tract. They do have sexual reproduction, and typically nematodes are either male or female and not hermaphrodites. And they do undergo internal fertilization. So free-living nematodes are very interesting, but we're going to go ahead and pull out two nematodes for particular mention and discussion that are parasites. And so they have a somewhat different and more complicated life cycle than many of the very simple nematodes. So we're going to consider two that specifically parasitize humans, the guinea worms. I'm just giving you the common name. I won't bother you with the species. And then the other one, Trichinella spiralis, which is another important parasite in humans. So let's go ahead and start with the guinea worms then. I thought this was an interesting group to bring up first because this is one of the few examples of important parasites or pathogens that has almost been eradicated worldwide. I'm making it a little bit darker just so you can, in here so you can see, hopefully. I grabbed this page from the Carter Center. This is a nonprofit that was started by former President Jimmy Carter. And one of the important international public health issues that they decided to tackle was guinea worm. And so in 1986, there were about 3 million cases. Virtually all of them were in Africa. This is the outline of Africa right here. I don't know if you can see it. There are only about 100 cases right now, primarily in the Sudan. So of course this part of the world is having political difficulties which interferes with public health efforts. But nonetheless, this is a rather extraordinary case of bringing down this dreadful parasite. So why is this so terrible? What is the life cycle like? This is a diagram from the Center for Disease Control in Atlanta showing the life cycle, this should make it easy to understand how the public health efforts have been able to knock down these numbers. Let's start by considering how people end up getting infected in the first place. That's shown over here, up here at the top, as step number 1. A human gets a guinea worm by drinking water that contains copepods, those are little tiny crustaceans that are infected with the larvae. That's it. This means you can stop the cycle right there if you either boil the water, killing off the larvae, or you just simply filter the water. You can stop the cycle right here. And this is a really unusual case in that humans are an absolutely required host for this particular life cycle. Which means if we can get to a point where not a single human has the guinea worm, then they have been completely eradicated. There is no other animal reservoir that can take the place of the humans. Okay, but if in fact the human does end up drinking this contaminated water, then what happens? The copepods die in the human's digestive system, but the larvae are fine. The larvae then go ahead and penetrate the walls of the digestive tract here, and then they mature and reproduce inside the body of the human. Then the male ends up dying, the male worms, and the female worm, by the way, there's a huge difference in size. I don't know if you can see that here. The male worms tend to be very tiny. The females are just huge, like this, very, very long worms, very high reproductive capacity. So then what happens is that the female worm, which is now fertilized from having sex with the male, then migrates to the surface of the skin, causes the blister to rupture, discharges the larvae. If this happens then in the water, as shown in the lower down here, then the larvae enter the water, can enter a copepod, and then nothing happens unless the copepods are then ingested by a human, because the human is the absolutely required host. So this is worse than the tapeworms we were discussing before, because they're not just staying put in the adult worm, tapeworms that is. These worms are not just staying put in the lumen of the digestive tract. They're migrating all over the body, and it's apparently very painful when they start emerging from your skin. And I have a picture here to show you what this looks like. Here is an individual's foot. This is the female worm being removed. They're very, very long, skinny worms, and so the best way of doing this without breaking them, because you want to get the whole worm out, not just snap it, is to wrap it around a cylindrical object like this. So sort of roll it up as it's being pulled out. So I think this is a very interesting case, because this is one of the very few cases in which this is a public health, an amazing public health success story and a triumph. And it is because of the life cycle of this worm that humans are required hosts, that we can just tackle this fairly easily. Let's move on now to trichinella, another nematode that parasitizes humans. And this is one that is cosmopolitan, and there is sadly little hope of eradicating it at the present time at all. Humans get this one, again, from eating improperly cooked meats, and so, again, it's a case of ingesting muscle tissue in which the immature worms have encysted. So this is a figure from your book, figure 34-26, showing the muscle tissue, and there's the juveniles right here. So this is what the life cycle looks like for trichinella, and it's an interesting comparison with the guinea worm. So if a human ingests undercooked meat, regardless of the source, then again you have larvae coming out in the intestine, moving around in the body, and then encysting in the human's muscle. So this is then a dead end unless something consumes the human muscle tissue. So you might think, well, why is this not a dead end? Because there are a lot of other animals, both domestic animals and also wildlife, that are reservoirs for the same individuals. So even if nobody consumes the human muscle and passes on these worms, there are other sources. So I'm going to go ahead and stop right there. We'll do the arthropods next Wednesday. Have a great long weekend, and I'll see you next week.